About some of the things that we've learned working on the spring snail group Pyrgulopsis over the last few years. Many of these species have been petitioned for listing. They're found throughout the western United States and south into Mexico. There's about 150 species in this group. Spring snails are pretty small, usually found concentrated around the spring head and spring runs in the west. They were usually thought to be endemic to a single spring or a spring system. But this gives us some challenges for taxonomic work, because if you are assuming that an organism is only found in a single site, then you don't really have an understanding of what population level versus species level differentiation might look like. And so this assumption that species tend to be found only in a single site has led to some difficulties with the taxonomic work. So I wanted to tell you a little bit about that. Some of the questions that we were trying to address, there's been a lot of barcoding data generated for these, and so there have been numerous distinct divergent mitochondrial lineages have been discovered, and we want to know if those should be described as species. And we also want to test the utility of some of the characters that have been used to distinguish these species in the past. I'm going to speak today about peniomorphology and shell morphology. Now, for peniomorphology, we kind of find a mix of utilities. So for example, this slide is showing you 3 species that we are proposing to synonymize into one species. On the right, you see the peniomorphology for these 3 species. They basically vary in the number and the extent of this terminal gland on the penis, from small, medium, and then large. What we found when we dissected and then characterized and then sequenced individuals was, for example, this population here at 9 Mile Reservoir in the blue. Each of the 3 peniomorphologies are present in those individuals in that population, and they all have identical CO1 sequences. We have a very shallow clade with peniomorphology being scattered throughout. It's not a diagnostic feature. However, in other species, it does appear that it can be useful. So for example, this clade over here on the right, lineage C, it has these large rounded glands that are ciliated at the end, several of them, every male has them, and they're really different from what you see in the closest relatives. And so in some cases, these can be diagnostic, but they're not necessarily diagnostic, especially little minor differences. Another character that we looked at is shell morphology. So in terms of shell morphology, Pyrgulopsis are just not that distinct in shell shape, and for the most part. So these 4 species on the left here, pretty similar in shell morphology. Occasionally you have 1 or 2 species that are pretty distinctive. When we look at shell morphometrics, what I wanted to show you here on this top graph, this is those same 3 species that we were trying to determine whether or not to synonymize them. And the polygons on this top graph, this is a least discriminant analysis. Each of these polygons is a population. And so what we see is that you can tell populations apart. So you can tell an individual that looks like one from Bear Lake versus an individual that looks like 9 Mile Springs, and they are different in shell shape. However, there's a population level differences. When we take all of these same individuals and compare them to other more distantly related species, everything from this top graph is encompassed in this polygon that's lineage A and different in shape from other good species. So the shell morphology is easily confounded by this single site issue. Some populations have a distinct shape, but in examining in conjunction with other data sets, we would assign these to the same species. They can become distinct in small local populations. So some of the lessons that we have learned, we can't assume all species are single-site endemics, and that being the prevailing wisdom has made the taxonomy more challenging. We've found that you may also have more than 1 spring snail species per site, and you may not be able to tell them apart. Sometimes you can have members of different genera that are very hard to tell apart at the same site. So what this means is when you're trying to work on them, you have to document all of your morphological and genetic features from the same individual. You can't just go collect 20 and then send 5 for DNA and 5 for radula and 5 for shell shape, because you might be collecting those data sets for different species. So you have to document morphology before you destroy them for DNA work. We can't just rely on any single character and assume it's diagnostic, and we really need genomic data to tell species apart in this group. Just CO1 we've reached the limits of what that can do. Okay, so one last point. I just got some funding from Texas Parks and Wildlife to go sample a couple hundred more springs in the mountains in West Texas. So if you know any potential people who are looking for a master's degree who think that that would be a lot of fun, please have them send me a message. Thanks, and I look forward to taking your questions online.